Good afternoon, students. In the previous class, you have learnt about nanoparticles, the method of preparation like top down approach and bottom up approach, and then their advantages. Okay. Today, let us see characterization of nanomaterials. Okay. Now, there are three analytical tools to characterize nanomaterials. Okay. Now, which are those? Scanning electron microscopy, in short or in brief it is known as SEM. Transmission electron microscopy, in short it is called TEM. And the third one is X-ray diffraction. Okay. Now, in this, among these three, scanning electron microscope is most widely used all over the world. Alright. So, this we are going to study in detail. Okay. Now, scanning electron microscopy, that is SEM. The scanning electron microscopy is one of the most widely used instrumental method for nanomaterials categorization. It is a type of electron microscope that images the sample surface by scanning it with a high energy beam of electrons. The electrons interact with the atoms that make up the sample, producing signals that contain information about the sample's surface topography, composition and other properties. Okay, now what is the summary of this? So, scanning electron microscope is nothing but a type of microscope which will produce images of the sample surface. Okay, by which method? By scanning. Now, how it scans? In this microscope, a high energy beam of electrons are passed, which scan, they collide with the atoms of the surface, okay, and then they scan. So, the electrons interact with the atoms and hence they produce signals, and these signals, okay, give you the information about sample surface topography, composition and properties. Okay. Next slide. The scanning electron microscope, that is SEM, is an instrument that produces largely magnified image by using electrons to form an image. Okay. By making use of electrons, we can produce what? Magnified image of the sample. Okay. Magnified image of the sample. So, this method is very important method. A beam of electrons is produced at the top of microscope by an electron gun. How a beam of electrons is produced? At the top of the microscope there is a electron gun which produces what? A beam of electrons. Okay. Next slide. Electron beam follows a vertical path through the microscope which is held within a vacuum, travels through electromagnetic fields and lenses which focus the beam down towards the sample. Okay. Electron beam gun produces electron beam which is focused in the electron microscope. Okay, that means it is made to fall on the surface. Okay, so hence here we get what? Magnified image. Detectors collect these X-rays, backscattered electrons and secondary electrons and convert it into signals. Okay, detectors convert this, okay, X-rays, backscattered electrons and secondary electrons and convert it into signals that is sent to the screen, and those signals are sent to the screen, okay, similar to TV screen. Alright. This produces what? Final image. The examinations are generally made on the photographic records of the screen. So, thus we get what? A magnified image of the sample. Okay. Now, here there is the diagram. Okay. Diagram it shows what? The SEM. You can see here electron gun here, which produces what? A beam of electrons. This is the beam of electrons. Okay, and there is an anode here. There is magnetic lenses here. Okay, it is passed through scanning coils. You can see scanning coils over here. Okay, and then this is the back scattered electron detector, and then there is a sample here. Specimen is kept on the stage. Okay, and the secondary and backscattered electrons are detected here and will go to a TV scanner, okay, which will produce image. So, a TV screen you will get a magnified image. Okay. Now, what are the essential components of all scanning electron microscope? Include electron gun, which I have already shown here, electron gun, electron lenses, okay, electron lenses are nothing but magnetic lenses here, sample stage, that is a stage or place where you can keep the sample, okay, or specimen, detectors for all signals, you can see the detectors over here, okay, for the signals, and display or data output device. So, display will be done here. Okay. Next slide. So, this is another diagram, or I can say cross sectional diagram of scanning electron microscope. You can see here electron gun, which is actually inside the microscope. This is electron gun. This is cross sectional diagram. Vacuum chamber is this. Okay. Anode is here. Condenser lens are here. Okay. Electron beam is passed, okay, through what? Electron, from the electron gun through this anode, etc. Okay. Condenser lens is here, objective lens is here, okay, sample chamber. This big chamber is the sample chamber where you have a X-ray detector, sample is placed over this specimen is placed over the stage, okay, secondary detector and then backscatter detector, okay. This is another diagram, we show the representation of SEM, okay, you can see electron source, anode, condenser lens, objective lens, backscatter detector, X-ray detector, secondary electron detector, sample you can see here down, okay. So, this will be generated here, scanning is done here and then it will be shown in the TV screen, okay, in the magnified image will be shown over here, alright. Next. Why SEM is preferred over other microscope? Okay, other microscopes to characterize nanoparticles, or what is the advantages of SEM over other microscopes? Okay, large depth of field which allows more of sample specimen to be in focus at one time. You can focus the electron beam on the specimen so that large focusing is done. Okay, so large depth of field, okay, which allow more samples of the specimen to be focused, so that focusing is done, okay, to a greater extent. Then high resolution, so closely spaced specimens can be magnified at much higher levels of magnification. So, high resolution means high level of magnification is done, okay, in this microscope. Control over magnification, you can control the magnification also, okay, and then you will get what? Clear image of the sample, okay. So, these are the advantages of SEM over other microscopes, okay. Next slide. Preparation of nanoparticles by chemical method. Now, how do we prepare nanoparticles by chemical method? Okay. So, sol gel method is one of the chemical methods, okay, to prepare what? Nanoparticles, okay, sol gel method. Sol gel method is one of the novel method to produce fine particles with narrow side distribution and control chemical composition at comparatively low temperature. Okay, you know that gel is one of the colloidal system, correct? Sol is nothing but colloidal solution. Okay, gel is one of the types. This method is, that's why it is called sol gel method. So, you get one at a lower temperature, you get particles of this size, nano size, okay. So, that is why method is called sol gel method, by making the solution into to be get converted into gel form, okay. In this method, dispersion of the particle of the metal, you know that in a colloidal solution there will be two phases, disperse phase and dispersion medium, okay, and based on that the classification of colloidal system is done. So, in this process, dispersion of the particles of the metal compound usually in aqueous phase is first prepared. Okay, you should prepare the aqueous phase dispersion as dispersion medium. So, particles are to be dispersed. Okay, medium is aqueous phase, and then converted into gel particles. Okay, so this is one of the types of colloidal solutions. Gelation is achieved by means of chemical dehydrated agent with the surfactant 2-ethyl cyclohexanol, which is the surfactant used 2-ethyl hexanol. The particle size is achieved by means of by the precipitation state is maintained during sol and gel. So, you will get the particles of colloidal size or nanoparticle size. Okay. So, this process is better control on particle size and size distribution. Now, the sol gel process is a wet chemical technique also known as chemical solution deposition and involves several steps. in the following chronological order: hydrolysis and polycondensation, gelation, aging, drying, densification and crystallization. Next slide. Now, what are the advantages of nanomaterials? Okay, so you know that nanomaterials have got wide applications. Okay, so what are the important applications of or advantages of nanomaterials? First one, nanocrystalline silicon carbide is a candidate material for artificial valves, primarily due to its low weight, high strength, wear resistance and biocompatibility. Okay. So, nanomaterial, nanoparticles are used in the preparation of artificial heart valves, okay, which is that silicon carbide. So, silicon carbide is converted into nanoparticle size and it is used in the manufacture of artificial heart valves. Okay. What is the reason? It has got low weight, high strength and wear resistance. Okay. It will not tear. So, it has got high strength and then biocompatibility. Okay. So, it will matches or fit into our human beings. Okay. So, as heart valves. So, it is widely used. Second one, fullerenes. Okay. So, in the manufacture of fullerenes, which is nothing but carbon 60 allotropes of carbon. So, this is converted into nanoparticle size. Okay. So, which are graphene sheets rolled into tubes or sphere. Okay. So, these are used as superconductors by which you can attain lower temperature 10 to 40 Kelvin. Okay. Third one, carbon nanotubes are used as memory devices. So, these are also used as what? Memory devices. Fourth one, nano ceramic filters are used for water purification. So, nanomaterials are also used in what? Water purification. These filters are mixtures of nano alumina filter and microglass. So, in the water purification, nano ceramic filters are used. Okay. What is that filters contain? Nano alumina filter and then microglass. Okay. Next slide. Nanomaterials are used as also used as what? Antioxidants. Then sixth one, they are also used in nanocomputers. Nanoparticles are used in nanocomputers as logic gates. Okay, so the molecular switches have to be connected together to form logic gates, which are used in nanocomputers. Next one, so to improve seed germination, okay, then growth, plant protection, pathogen detection, pesticide and herbicide residue detection, these nanomaterials are used. Okay. Then they are also used in gene transformation. Okay. So, they are used in gene transformation, drug delivery system, okay, and drug delivery system in order to improve therapeutic values. Alright. The ninth one, nanotechnology is also used to destroy cancer cells. So, nanomaterials are used to destroy what? Cancer cells. Okay, with a highly targeted package of tumor boosting genes without leaving any secondary effects on the body. Okay, so these will burst only what? Tumor cells. Okay, without what? Damaging the other tissues. So, good tissues will not get damaged. Okay, or second we can prevent what? Secondary effects on the body. Okay. Next. So, with this part is over. Okay. This chapter is over. So, any difficulty please contact me. Okay. Contact me. Thank you.